All right, so let's get started. It's my pleasure to introduce Ryuichi. Ryuichi received a BS in bioinformatics from the University of Tokyo in 2024, where he studied bacterial genome editing and MGE-driven dissemination of antimicrobial resistance genes. Ryuichi joined the CREST lab as a PhD student in August 2024, where he is investigating horizontal gene transfer mediated by mobile genetic elements and their evolutionary history. Outside the lab, Ryuichi loves to hike, cook, draw, sing, and read novels and mangas.
Okay, thank you so much for the kind introduction. So, hi everyone, I'm Ryuichi from Comparative, just like he mentioned. So today, I'm super excited to talk about my favorite topic in my life, which is how can we prepare, build, interpret, and love phylogenetic tree. Oops. So before, hmm, wait, sorry, I lost my. Okay. Before moving to the main talk, let me start from the acknowledgement. So first, shout out to the Brady and Jack and CCB team who is organizing this seminar, and my friend in Lubin and Kresan. Thank you so much. And also, huge thanks to Dr. Moto Matsui from Kyoto University who gave me the feedback on this presentation material. So here, I put the QR code of his personal website. So if you're interested in his work, including this novel method to build tree based on the similarity graph, please check this QR code. Okay, so let's move on to the presentation. But guys, please interrupt me anytime during this presentation. So the other day, one of my friends said to me, think that, hey, Ryuichi, phylogenetic tree pissed me off. Because I was like, why? And she said, because it doesn't tell me anything. And people will build phylogenetic tree when people want to show they have something to show to their PI. And I was like, wait, that's 50% true. Only 50% true because phylogenetic tree is juicy. You can learn a lot of things from phylogenetic tree. So here I put my favorite tree, which is the famous Banfield tree. So basically, this is tree of life, which means each tip, each leaf correspond to the one species. But of course, it is impossible to put all species in this world into one tree. So in this tree, one leaf contains multiple species. But the first thing, as a microbiologist, I am super excited by this tremendous diversity of bacteria. You know, this is species, this is all species in this world. Look at this tiny green clade. All eukaryotes, including ourselves, will fall into this tiny clade. Of course, we already know that eukaryotes are really diverse, including myself, ourselves, fish, fungi. But it's just that the diversity of bacteria is just breathtaking. So if you zoom up here, oh, please grab pizza. We have hot pizza right now. Yeah. So if you zoom up a tree like this, you will find a clade like this. Human comes here, and as a sister clade, there is bonobo and chimpanzee. So what does this mean? Okay, so important point about phylogenetic tree is where internal node, which means where two branches merges to each other, we assume there is hypothetical common ancestor of these two branches. Okay, so in this example, there was one hypothetical common ancestor species, which is blue species. And from this species, human evolved. And on the other hand, the common ancestor of bonobo and chimpanzee evolved from this species. And after that, they became bonobo and chimpanzee. Okay? So, of course, this raises one question. So I just said we can trace back to its ancestor from the tip of tree. If you keep doing this, if you keep tracing back its ancestor, can we, can we assume or can we reach to the ultimate ancestor of all life in this world? So, the answer is yes. People already tried that. So in this figure, they built very severe phylogenetic tree of all bacteria in the world. And very carefully, they try to identify the ultimate ancestor of bacteria in this figure, root. So according to this analysis, the root, which is common ancestor of bacteria, will be somewhere around here, near Fusobacteria. Based on this information, they could reconstruct the characteristic of common ancestor of all bacteria. So this is a proposed phenotype of all bacterial common ancestor, which is rod-shaped cell, has double membrane, and can move around, and can sense chemical signal around them, which is quorum sensing. Like, for me, the most surprising thing is like they have CRISPR-Cas system. So, I was like, I didn't know that. Oh wow, CRISPR-Cas is really ancient system because even the ultimate ancestor of all bacteria possess CRISPR-Cas system. So, of course, this is just so fun to know. But also, this points out really important biology, which is even in the era of common ancestor of bacteria, there was a phage. Because CRISPR-Cas system is a system to fight against phage. The fact that CRISPR-Cas is that ancient means, reflects the long coevolution history of bacteria and phage. So, it was a really good example of phylogenetic tree itself being cool science. But phylogenetic tree can also be a good starting point or a good tool to do cool science. Okay, so let's assume you're interested in long-living phenotype of some animal or something. And you want to find a gene which is responsible for long-living phenotype. And I think you will just do something like enrichment analysis of gene, focusing on the long-living species. And let's assume you already identified gene A is enriched in long-living species. Of course, you'll be super excited. But if the distribution of species with gene A and long-living is like this, what do you think? I will be concerned because, judging from this tree, there's also a possibility that, these three species, their common ancestor was, somehow long-living and somehow possessed gene A. And because of that, all species derived from this ancestor is showing the same phenotype. In a case like this, can we say we identified three distinct species which possess gene A and long-living? Maybe not. This is good example of phylogenetic signal is biosingular result. If you actually want to find a gene which is responsible for long-living phenotype, you want to find gene like this, distributed in long-living species and scatter across the species tree. If you could identify gene like this, now you can build confidence that, okay, maybe this is bona fide gene responsible for long-living phenotype. Okay, so, there are so many motivations to build phylogenetic tree. I just introduced two examples, but of course there are more. But before that, I think today, I want to talk about more practical stuff. So, in my opinion, why, oh, sorry, why so many people dislike phylogenetic tree? In my opinion, because they look complicated. But they're not. Once you know how to read tree, this is really crystal clear. So let's talk about how to read the numbers put on the tree. Okay, so first, as I mentioned before, every internal node assumes hypothetical common ancestor of each leaf. So in this example, from this common ancestor is the ancestor of species 4 and 5. From this ancestor, species 4 and 5 emerge. And this number reflects the evolutionary distance from their ancestor. In this case, both 4 and 5 are 0.1 distance away from their common ancestor. Evolutionary distance sometimes means just number of substitution or mutation accumulated. The other time, that means actual time. Okay? So, same thing for all internal node. Okay, let's talk about one more example. Let's say this green species this green species is the ancestor of species 1, 2, 3. So from this green species, species 3 emerged with distance of 0.2. And on the other hand, the common ancestor of species 1 and 2 also emerged from this guy. After that, this purple species became species 1 and 2 with this distance. Okay, it's really easy. This branch length simply reflects the evolutionary distance from its ancestor. Okay.
Like,
Mhm.
is that kind of internally normalized? So like within this tree, 0.2 is double 0.1,
Mhm.
but 0.2 in a different tree would be like a different scalar.
That's really good question. You know, for example, in this tree, all of the tips are aligned on the same line, right? But no, if we think about coelacanths, some species still possess ancient phenotype, which means they are really close to their ancestor. If you want to build actual tree, some species which still look like its ancestor will be located somewhere right here. So when I align all of the leaves like this, I already needed to have some assumption, some calculational assumption, including the one Jamie pointed out. But yeah, there are so many other ways. Because of course, some branch, some clade will evolve faster than other clade. We also want to consider such difference distribution. But in many cases, we cannot have ideal tree. We need to have some assumption to build tree, even if the assumption may make my tree a bit far from practical tree. But yeah, thank you so much. So then let's think about this number put on each clade. So this number is called branch support, which means how reliable each branch is. So, but first I was confused. Hey, what do you mean by how reliable each branch is? So, if you have 5 species, there are so many ways, so many possible trees to explain the phylogeny of these 5 species. Here I put a tree like this, but also other tree like this is possible. Let's think about this tree. In this tree, the green circle, the 98% circle, square is maintained because species 1, 2, 3 are under the same branch, same clade. But not for red square because species 1 and 2 are not under single same clade. So, considering all possibilities like that, how reliable each clade is. I'm going to talk about how to calculate this later, but this is the concept of branch support. So, okay, so let's talk about actual procedure to build phylogenetic tree. When you build a tree, the process will consist of these 4 steps: sequence alignment, model selection, tree inference, and assessment. So first, I'm going to talk about sequence alignment. What's alignment and how can we do that? Okay, so regardless of your purpose, you will need to prepare sequence dataset if you want to build a tree. Today, I'm just going to talk about how to build species tree of life. So in a case like this, I need to find marker gene for each species. I want marker genes to be different species to species because I want to distinguish different species in sequence level. So in such purpose, 16S ribosomal RNA is the kind of gold standard in this field. But other people, including Professor Banfield, uses ribosomal protein, or of course, other marker genes exist and are a good approach also. So in this figure, each square can be DNA, RNA, or amino acid sequences. So after you prepare, Mhm.
Is there any reason to choose the different genes?
Mhm.
Any
Yeah.
the one over the other or something?
So, for example, if you want to build the first tree I showed, the huge, massive bacteria eukaryote tree, sometimes 16S ribosomal RNA doesn't contain enough information, to cover the true diversity of all species in the world. But in that paper, what people did is like they concatenated all ribosomal proteins. So, of course, concatenated protein is much longer than just one protein or 16th ribosomal RNA. Because they concatenated marker genes, they have longer sequence. Longer sequence has more information to distinguish each species, even in such a huge clade, even in such a huge tree. If you are bacteria, if you're a microbiologist, and if you're interested in comparative genomics, I think you have heard GTDB, which is a huge database of bacterial genome. GTDB constructed phylogenetic tree of 160K, 130K, yeah, bacteria. How they did is like they concatenated so many marker genes, marker proteins, to get more information to infer actual phylogeny. So if you want to build huge strict tree, you want to take such approach instead of just 16S.
So if you take a nucleotide approach versus
Mhm.
an amino acid approach, does the nucleotide approach give you maybe more accuracy because it takes into account like wobble-based things?
That's a great point because nucleotide mutation will just result in the same amino acid in many cases, right? So if you take marker genes and if you want to build phylogenetic tree, nucleotide approach is more practical or more severe sensitive. That's 100% correct. But in many cases, for example, if you're interested in some protein family recombinase, if you just want to find subfamily in this protein, just use amino acid. It's fine because if you're interested in protein, the protein sequence difference matters, but nucleotide sequence diversity doesn't matter. So if you're interested in protein tree, just use amino acid. Thank you. Okay. So after preparing sequences, you will need to align the sequences so that corresponding region will be in the same column like this. This process is called alignment. There are so many softwares to do this, but I personally really like Maft or Muscle T-Coffee is also great. Important step after alignment is this trimming. If you align like 1,000 sequences, you will often encounter region like this. Region which are poorly aligned or gappy region. Regions like this don't contain so much information. So this will slow down the subsequent analysis. They don't have sequence information, but the overall sequence gets longer, or the subsequent analysis will be slower. And also, sometimes such trash region can just be harmful in the subsequent analysis. So usually people just trim the sequence based on how well this region has been aligned. But one caution here, if you trim too much, you will lose too much information. So in this example, because we trim too much, we can no longer distinguish species 4-5 or species 2-3. This happens. So if you trim, please be careful. So the software including Trimer, Cam Cleaner, ClipKit, but just use whatever makes the most sense to you. I use ClipKit. So after alignment, you will need to select evolutionary model before moving to the tree inference. But don't worry, this is usually automatic process in your software. So we will not do this manually. Your software will do this manually or automatically. But let me just introduce the concept of evolutionary model. So for example, in nucleotide sequences, the mutation rate from A to G, mutation rate from A to T is completely different. Because A and G, both of them are purine, and T and C, both of them are pyrimidine. Transition is more likely to occur than transversion. Okay? So, we cannot overlook this big difference when we do some, calculation based on mutation rate substitution. So this is one simple approach. We can just treat them differently. We can just have one or two parameters to treat the mutation rate of transition and mutation rate of transversion differently. Okay, this is really simple. We are just saying that let's treat transition transversion differently in subsequent analysis. But this is the essence of evolutionary model. Okay, of course, there is so many other evolutionary models, but I just showed one of the most simple ones. Same thing for amino acid substitution, but there are 20 amino acids. It's really hard to build a substitution model based on some parameter like this. So people are just using empirical model inferred from the existing protein database. But this is the concept of evolutionary model, really easy. Just treat different substitution differently. Okay, so after that, finally, we can do tree inference.
Just to clear up, like, how exactly does model selection work? Is it just like, does it have like a bunch of matrices that correspond to these different evolutionary models, or does it dynamically learn them, or like
So-
how do you have multiple of these to pick from?
Yeah. So for example, if you use software called IQ-Tree2, there is a software implemented which is called Model Finder Plus. In the Model Finder Plus, IQ-Tree will do very fast but very crude tree inference from your input using different model. Okay, there is like 1,000 models in IQ-Tree. IQ-Tree will use every single model. And using every single model, IQ-Tree will do really crude but really fast tree inference and calculate some score, how good each tree looks like. And IQ-Tree will just pick the model which resulted in the best tree in the crude analysis. That's how model finder works. And if your question is about why there are so many different models, because now, for example, our mitochondrial DNA or the genome of HIV or some SARS-CoV-2, all of them have completely different mutation rate. So if you are interested in mitochondria, and if you want to build a tree based on mitochondrial genome, you want to use substitution model specialized for mitochondria. Because of such needs, there are so many evolutionary model. So before moving on tree inference, what I want to emphasize is, even if you have only small number of leaves, the number of all possible tree is massive. For example, let's say you only have 5 species, but the possible topology is 15. And for each topology, there are different ways to align leaves for each tree. So, just the number of all possible tree is huge. So the process to find good tree looks like this. In the space of all possible trees, let's say this is a good tree. You want to find this one, exactly this one. Or, maybe let's start from somewhere in this space. You want to find a way to reach to good tree somehow. So how can we do that? Of course, we need to use some metrics. If we could have metrics which will show higher value for beta tree, you can draw a space like this. And we can just climb up the mountain of metrics, aiming for the beta tree in this space. So of course, there are so many ways to define metrics. But today, I'm gonna just talk about maximum likelihood method, which is the most commonly used, and in many cases, the best metrics to do tree. So maximum likelihood This is really simplified figure, but it captures the essence, I believe. So how does maximum likelihood work? As I mentioned before, we can infer a tree using sequence and model, right? There is some, cool calculation method which can do kind of opposite thing. If we use the calculation method, we can use tree and model and calculate the probability that these two things will spit out this input sequence. Okay? The calculation will calculate the probability that this input will emerge from this tree and this model. This is the opposite thing as we were discussing before. It's just like this. Let's say we use tree A and this model to calculate the possibility of this input. And it was like E minus 100. But if you use tree B and model to calculate the probability of this input, it was much, much higher. In this case, we can say, okay, tree B is much more likely to be the correct tree. Okay, this is the essence of maximum likelihood method. So after building a tree, there's still one more really important step, which is assessment. As I mentioned before, on the tree, there is a branch support value, which means how reliable each clade is. Assessment is a process to calculate this value. Of course, there are so many ways to calculate branch support, but today I'm going to talk about the most commonly used method, which is bootstrap value. So if you calculate bootstrap value, it will be like this. First, you will do random sampling from your sequence alignment. And you will infer a tree only based on this randomly sampled sequence. You can subsample from the same column multiple times. But the important thing is like iterate this entire process again and again for like 500 times, ideally 1000 times. So this is how bootstrap value works. Let's say we did this iteration for 100 times. And after 100 iteration, let's say this tree obtained 53 times, this tree 40 times, and so on. Okay, let's get back to the original tree. In original tree, I said the reliability of this clade is 60%. Because after 100 iterations, this red square was captured 60 times in total. This one and this one and this one. So we can say in the process of bootstrap, in 60% of the case, this clade was reproduced. Okay, this is the concept of bootstrap. For example, this blue clade, 95%, really high. It makes sense because this clade 4 and 5 is almost always almost always captured in the process of bootstrap value. Okay, so this is how we calculate bootstrap value. And okay, as a procedure, that's pretty much all I wanted to talk about how to build a tree. So now how does it look? It used to look like more plain, more boring, but now it looks more, it looks like contains more information than before, right? So I really hope today's talk will help you to infer tree on the paper or will motivate you to build your own tree. Thank you so much. So is there any question to discuss?
I have a question. So, in the maximum likelihood,
Mhm.
how is like the tree inputted as a value? Because I'm assuming it's just like numeric calculations to get the input alignment, like probability output.
Mm-hmm.
So how do you like quantify a tree as like a numerical value?
Mm-hmm. Yeah, that's really good question. For example, then I should have put a label on this, but okay, let's say this is species 1, 2, 3. Okay, and this is species 1, 2, 3. Species 1 and 2. Species 1, 2. This column is different, right? 1, 2, 3. Okay, this is species 3, 2, 1. Only species 1 has pink in here, right? Which means species 1 underwent some mutation from orange to pink. And then, okay, let's look at the model because your model will tell you the probability or mutation rate of all possible mutation. If your model says, hey, in my model, mutation from orange to pink is impossible. In a case like this, we can say the probability that this input sequence is 0, right? Because we assume this tree and this model is correct. But if we assume that, we need to assume the mutation from orange to pink, which is impossible in this model. In a case like that, we can say, okay, probability is 0. Okay, calculation like that, if we do calculation like that for all column, for all possible pair, we can calculate the entire probability of this input sequence. This is really complicated process, but I think the essence is like that. It was clear.
Yeah,
Thank
that's
you. Yeah.
You know, like the people that are doing the bigger trees that
Mhm.
are trying to like identify Luca,
Mhm.
like in some of these like maximum likelihood calculations, is there like kind of like a quotient or something for horizontal gene transfer?
Mhm.
Because like here the assumption always is like if two things have something, then their shared ancestor also
Mhm.
had it.
Mhm.
Like is there like a probability that you like sneak in there?
Mhm. Yeah, that's really good question. When people talk about horizontal gene transfer, like let's say this is species tree, right? You can also build protein tree based on the gene of interest. If the tree of protein looks exactly like this. Oh, wait, I can draw, right?
Yeah.
Thank you.
You want to turn on your camera?
Oh, yeah. Can you?
Oh, on your screen. So I can see it.
I'm gonna. Okay. Sorry, some time card. Okay, let's say this is a species tree you drew. Species tree. And if your tree of protein you are interested in looks exactly the same, let's line the corresponding one. The two tree will look like this. If you obtain a result like this, you can say, okay, maybe this gene didn't undergo horizontal transfer. But if you got completely different tree, oh wait, I don't know how to make a completely different tree. Okay. If you got completely different tree, and if you line corresponding thing, like something like this, if you got plot like this, now we can build confidence that, okay, maybe horizontal gene transfer happened. Maybe this protein transferred to this species. This is called co-file plot. So there is so many ways to discuss about the horizontal gene transfer, but the point is this co-file plot assumes correct species tree and correct gene tree, right? The point is there's possibility if you build a species tree based on some marker gene, And if this marker gene somehow horizontally transferred, that will completely ruin your species tree for sure. So the precaution here is like when you choose your marker gene, please make it sure that this gene is really unlikely to be horizontally transferred. But this is kind of like the only approach to deal with the possibility the horizontal gene transfer ruin species tree. Thank you. Yeah, thanks.
So people sometimes incorporate like time on
Yeah.
y-axis, so is that kind of like, is it strictly, it's like, a distance based on like, nucleotide differences, and then that translated to like mutation rates based on your model? Is that kind of how that works? Or, yeah.
That's correct. If you want to say evolutionary distance corresponds to actual time, you need to put really, really strict assumption, which is the evolutionary speed or mutation rate is identical in the entire tree. For example, if species 4 and 5 are high mutator, if species 1 to 3 are not high mutator, this assumption will not work. We can say evolutionary distance equal to actual time only when all species, all clade are evolving in identical speed, which is really unlikely to be the case. So, I think if you got a tree, basically we can think that your tree doesn't reflect actual time. Only when you treat your data carefully, you choose your protein marker gene carefully, only when you fulfill all requirements to do that, you can assume, okay, evolutionary distance equal to actual time in this tree. Yeah. Thank you.
And when you had your alignment for like
Mhm.
gaps, if you wanted to include those gaps when you're building your tree,
Mhm.
can you like, I don't know, put that into your evolutionary model?
Yeah, that's really good question. But basically, evolutionary model will only account for substitution. If you want to consider gap, that will be that will more likely to be the work of multiple sequence alignment. I mentioned there are so many ways to do multiple sequence alignment. For example, math treats gap in very unique way. Other software treats gap different way. So basically, one simple answer is gap will be considered when you build multiple sequence alignment. But sometimes this kind of relation is pretty informative. For example, some family has domain insertion in here. We want to consider that, right? So in a case like that, one, I think we want to be careful about trimming. And two, we can also utilize structural information. So tcofee is such software. tcofee can consider both structural similarity and sequence similarity to build sequence alignment. So, of course, it depends on your purpose, but there are so many ways to extract information from such gap illusion.
What work has been done for building phylogenetic trees off of like functional annotations for like the functions of genes?
That's really a good question. Because phylogeny doesn't always correlate with function, right? So very similar sequence can do completely different thing. Or because of convergent evolution, completely different sequence can do the same thing. So the short answer is no. There is no method to build tree based on the functional annotation. But there is an approach to build tree using protein language model. So protein language model, if you feed your sequence, I think Jack can explain this better. If you use protein language model, this is your input sequence. Protein language model can spit out some vector, embedding vector. And people assume that since protein language model is really great, this embedded vector reflects its function really well compared to the raw sequence. There are some people try to build a tree using this embedded vector. So in a sense, this kind of approach considers function similarity rather than sequence similarity. Thank you. Zach.
Would it be possible to try to infer like maybe missing leaves of your tree,
Mhm.
something like that? So,
Ooh.
things.
Yeah, I don't know the short answer, but that's really good point. Because if there is missing tree or missing clade, I think the overall reliability of your tree will be, much less than the actual tree, right? So I think, okay, in my understanding, if you have big missing clade, big missing leaves, let's say, actually there is one more protein here. If this is the case, I think bootstrap value around here will be much lower. Of course, I can be wrong, but this is my understanding. There is no direct way to assume, okay, maybe some leaf is missing, but we can assume maybe structure around here is not reliable based on the bootstrap value. Okay, cool. Okay.
Let's start.
Mhm.
How would you build a tree of like bacteriophages where some bacteriophages might not have any common genes?
Yeah, that's a good point. So the standard practice is like many bacteriophages has gene called terminator. So people, if you really want to do sequence tree using phage, I think you will just use terminator sequence. But of course, there is so many weird phage which don't even have terminator. In a case like that, this is impossible to build a tree, but instead you can build network. Network like put similar phage in similar location in this network. It's a different family of phage. Some people build this kind of network using proteome similarity, like how many genes two phage share to each other. This kind of information is really robust. Even if phage is lacking some genes, the overall proteome similarity can be calculated. So this is called V-contact. If you want to think about the subfamily or superfamily of phage, proteome similarity network is really robust approach. Yeah.
I guess in practice, do they combine those two techniques where they look at marker genes and the proteome similarity, or is
Yeah.
it like completely separate?
So as long as I know, there is no software which combine these two strategies, because these two strategies kind of like complementing each other. Proteome network is really powerful when your phage doesn't have marker gene, something like that. If your phage has shared marker gene, you can just do sequence similarity. But there can be a way to combine these two approach. Like first, maybe construct similarity network based on proteome first. And from this network, for the phage which has marker gene, you can also build phylogenetic tree to do more severe discussion on its phylogeny. So if we want to combine these two approaches, I think a way like that can be possible. Jack.
All right, two things. Could you remove the blur from your background on Zoom so you can see?
Yeah, I'm sorry. Thank you. I don't have blur. But
It's blurring your background
yeah,
a little bit.
I think-
Can't tell if it's just like, I don't think it's just low res.
my MacBook camera is really bad.
Yeah.
Yeah.
Well, it looks like it's isolating you.
Okay. Yeah, true.
I mean, I
Okay. But it should be fine. And second
But
thing.
yeah, other question is, so if you're trying to reconstruct something, I can't remember the details of this paper, like how they actually like infer genes, but does that involve something like, like with like the contact like where you make an ortholog table of all the different like proteins that are functionally related, and then you like try and like reconstruct an ancestral state out of that table, or right, like I'm just like kind of confused, like if you have some sort of tree that you made based on marker genes, how do you like go back and reconstruct the ancestral state that goes beyond just whatever marker genes that you have to reduce to make the
That-
generation of the tree tractable?
That's a good question. Ancestor state reconstruction is really deep field. I wish I knew more about that. But okay, there is one software kind of gold standard to infer the ancestor state, which is called PassML. PassML. So it also uses maximum likelihood to infer the ancestor state. So if I understand correctly, I think this guy is just explore the possible ancestor sequence or possible ancestor, phenotype, whatever, and calculate the likelihood of each possibility and just pick the highest one. But yeah.
But isn't that just gonna be like, because when I think of like ancestral state reconstruction, I think about like reconstructing the actual sequence that you like fit into it. So like usually we're like, right, if we're making marker genes, like, okay, you could construct the ancestral state of that marker gene, but if they were like in the Luca paper, they were going like, oh, okay, like we found that this has like CRISPR and then these like metabolic enzymes and so on and so forth. And I assume they didn't make a tree out of all of those, right?
Yeah.
If you don't know, that's fine.
Yeah, I'm gonna know. I will know.
Okay.
Thank you. All right. Is
All
there
right, well, if you don't have any other questions?
Thank you. Thank you so much, people on Zoom.
You got like bring your laptop up to the thing just in case.
Okay, okay.
Just
Oh,
for-
no, there is nobody. Oh, only Brady.
Well, we're gonna.
Yeah. Oh, oh, okay.